Hi students, good morning. Welcome to biology class. In the last sessions, in chapter 5, Principles of Inheritance and Variation, we have learnt about Mendel's laws of inheritance of one gene. Under inheritance of one gene, we have discussed about Punnett square, test cross, laws of inheritance, that is law of dominance and law of segregation, and also we have learnt about incomplete dominance and codominance. Today in this session we are going to discuss about inheritance of two genes. In the previous classes we discussed about inheritance of one genes. Mendel, he crossed plants having a pair of contrasting characters, like a tall plant is crossed with a dwarf plant, or a plant having violet flower was crossed with plant having white flower. In this way, he crossed plants differ in only one characters. Now, Mendel crossed pea plants differed in two characters. There is dihybrid cross. This is known as dihybrid cross. He crossed plants having yellow seeds and round seeds. Yellow seeds one character and round seeds. Or color of the seed and then here shape of the seed. He crossed this yellow seed, round seed having plants with green seed and wrinkled seeds plants. And then after hybridization, after crossing, he got F1 generation. This F1 generation, all the progeny, they resembled, they had only yellow and round seeds. Yellow and round seeds. From this, we can find out that this yellow and round seeds are the dominant. And also in the previous session, I taught you what are the 7 contrasting traits Mendel studied, that you should know. You have to learn all the 7 contrasting characters, because when problems are given, you should know which character is dominant and which character is recessive. Okay, now yellow seeds and round seeds, yellow and round are dominant. So in F1 generation, we got yellow and round seeds bearing plants. And then this genotype, or let us see the genotype of the symbols. Genotypic symbol for the genes. Y, we can give the symbol Y for yellow seed, and then round or capital or round, and then Y, capital Y for yellow seeds, and then what is the recessive for yellow? That is green. So we should not give G, we have to give small y, the dominant one's small letter y. And in the same way for round seeds we can give, because this is the dominant one, so capital R. This symbol is for round seed, and then wrinkled is the recessive form of the round seed. So we have to give small r for wrinkled seeds, not W. Okay, in the recessive letters we should not use, only the capital letters and small letters of the dominant characters only we have to use in crossing. Okay, now let us see the crossing. Now the genotype of the parents, we can write the genotypes as capital R, capital R, capital Y, capital Y. See, this is the one parent. Yellow round seed and yellow seed bearing plants. So capital R, capital R, capital Y, because they occur in pair. And then it is crossed with green and wrinkled green, small y, and wrinkled small r, small r. So small r, small y plants. Then we get gametes like this. This one R and then one Y, because this is dihybrid. So we should have 2 alleles having 2 characters. And here also small r, small y will get gametes like this. This capital R, capital Y is the gametes of this parent, and small r, small y is the gamete of this recessive parent. These two gametes, they fuse on fertilization and they produce F1 hybrid. So F1 hybrid, these two, they fuse together and we get F1 hybrid. Capital R, small r, capital Y, small y, capital R, small r, capital Y. So one capital R is so round seed, and then one capital Y dominant. In the presence of dominants, the recessive cannot express. So only dominant characters are expressed. So round and yellow seed, round and yellow seeds. That is capital R, small r, capital Y, small y. This is the genotype of the F1 hybrid. So in F1 we get only the dominant characters are dominant character. Only the dominant character is expressed in F1 generation, like in monohybrid. This is the same like monohybrid. When F1 progeny is self-hybridized, he found out that 3/4 of the F2 they had yellow seeds and 1/4 green seeds, because yellow dominant. So in F2 generation, 3/4 yellow seeds and 1/4 green in the same way, that is in the ratio of 3:1. And also this round and wrinkled seeds also they segregated in the same ratio, that is 3:1. 3/4 were round and then 1/4 were wrinkled. So yellow and green pair, yellow and green colored seeds, that character segregated in the ratio of 3:1, and also round and wrinkled, they also segregated in the ratio of 3:1, like that of monohybrid. And but this is dihybrid. But these two pair of characters, they act independently, just like monohybrid. They act independently. That is the basic principle of law of independent assortment. He proposed, after studying all these experiments, Mendel, he proposed law of independent assortment. Here, what is independent assortment? Independent segregation. This, even though two pairs of characters, different characters are there, the separation of this and the formation of gametes are independent of the other pair. They act independently, or the pair of characters. Look at page number 79 in your NCERT book. Now, you see, here, this is the result of dihybrid cross, where two parents differ in two pairs of contrasting traits, round and wrinkled and then yellow and green. So he crossbreed a plant having round and yellow seed with wrinkled and green seed. This one character is the dominant one and this is a recessive character. And then the gametes formed were one from each character, one genes. So capital R and small r. And here gametes small r and small y. Okay, so two types of gametes are formed, and these gametes, they fuse together and they form F1 generation. In F1 generation, all the progeny, all the individuals, they had round and yellow seeds, because these two, they fuse together and they form capital R, small r, capital Y, small y. These two gametes, they fuse together and they form zygote. From that zygote, a new plant is developed, and it had capital R, small r, and capital Y, small y. And then R is present, so it is a round. And here capital Y is present, so it is yellow. So F1 progeny, they resembled the dominant parent. And after that, he self-hybridized, he did self-pollination of this F1 generation. And then this F1 generation, they produced gametes in this way. With the help of Punnett square, you can easily find out the combination of these gametes, combination of genes for gametes. Now you see capital R, small r, capital R, small, you write here, and then capital Y, small y here. And then these together, because two characters, they have to combine together. So two characters genes also should be there in the gametes. So one in each pair of genes. And then so capital R, capital Y, and then capital Y, small r, and then small r, small y here, and then capital R, small y. So four types of gametes are produced from this F1 generation. One will be considered as 4 will be considered as male, and then 4 will be considered as same, because selfing. 4 same types of gametes have same genes. All the gametes, they have same genes. So female gametes, and then these are all considered as male gametes. And when they fuse together, zygotes are formed 16, and they produce new progeny, that is F2 generation. When they combine together, all these gametes, they fuse together and they form zygotes, and then the zygotes develop into new plants. You see, this capital R, capital Y, and capital R, capital Y, they fuse together and they form capital R, capital R, capital Y, capital Y. And then this capital R, Y, it combines with the small r, Y, and then it develops into a zygote, capital R, small r, capital Y, capital Y. So in this way, all the gametes, they fuse together and they produce the F2 progeny, F2 generation. Let us see how many different types of phenotypes are expressed. Here you see round and yellow seeds bearing plants. How many plants they had round and yellow? 1, 2, 3, 4, 5, 6, 7, 8, 9. Totally 9 seeds. 9 plants they had round and yellow seeds. 9 have round and yellow. Here also this is phenotype, and then the genotype of the round and yellow seeds, they differ. Capital Y, capital Y, capital R only one, and then capital Y, capital Y, capital R, small r, capital Y, and then only capital 1R, and then small r. 2 types of phenotypes. For 2 phenotypic 2 gametes, they had this type of genotype. So in this way, totally 4, 5, 6, 7, 8, 9. 9 plants, they had round and yellow seeds, but their genotype differs. In the same way, next, yellow and wrinkled. How many yellow and wrinkled? Yellow color seeds and wrinkled. 1, 2, and 3. 3 plants, F2 generation, they had yellow and wrinkled seeds. So, and here also there are 2 types of genotypes. See, capital Y, small r. So this is yellow and wrinkled. Here only one capital Y is there, so it also will be yellow, and then this round r, small, they both are wrinkled. So 2 wrinkled seeds are there, and then one this also, here also yellow and wrinkled seeds. And then so genotypes, here also 3 types of genotypes in yellow and wrinkled seeds. So 3 seeds we had. And then here this round and green. How many round and green we had? 1, 2, 3 round and green. Here genotypes also differ. So round and green we had 3, and totally 16, 9, 10, 11, 12, 13, 14, 15, and then 1, 16. This is green and wrinkled, only one green and wrinkled. So totally we had 316 individuals in F2 generation in the ratio of 9:3:3:1. 9 individuals, round and yellow seeds they had, and 3 plants they had round and green, and 3 had wrinkled and yellow, and 1 had wrinkled and green. So how many types of phenotypes we had? We got 1, 2, 3, 4. 4 types of phenotypes. And how many different types of genotypes we got in dihybrid? 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 different types of genotypes we got, and then 1, 2, 3, 4 types of phenotypic individuals we got in F2 generation of dihybrid cross. So in dihybrid cross, we got 4 types of phenotypes: round and yellow, wrinkled and yellow, round and green, and wrinkled and green. 4 different types of phenotypes in the ratio of 9:3:3:1. And then this ratio 9:3:3:1 can be derived as a combination series of 3 yellow and 1 green, and 3 round and 1 wrinkled. This is monohybrid. Just like in monohybrid 3:1. This yellow and green, they also formed in the ratio of 3:1, and then this round and wrinkled, they also expressed in 3:1 ratio, just like that of monohybrid. Now let us find out in what ratio they expressed. This pair of characters separately they expressed in 3:1 ratio. Now we find out, first we will see this round and wrinkled seeds. Round seeds, how many round seeds we had? Here 9 round seed, and also here round 3. So 9 plus 3, 12. So round seeds we got 12 round seeds. And then wrinkled, see here wrinkled, here 3 wrinkled, and then here 1 wrinkled. So 4. So 4 wrinkled seeds and then round 12. So what is the ratio? 3:1. So they separated in 3:1 and they expressed in 3:1 ratio. In the same way, yellow and green, next pair of characters, let us see. See yellow seeds, how many yellow seeds we got? Here 9 yellow seeds and then 3 yellow seeds. So 9 plus 3, 12. So 12 yellow seeds we got. And then green, how many green? You see here 3 green and here 1 green, 4 greens. So 4 green seeds. So what is the ratio here also? 3:1. Here also the dominant is 3 and then recessive is 1. Here also the dominant round is 3 and recessive wrinkled is 1. So here also 3:1 ratio. Both the characters they expressed in F2 generation in 3:1 ratio. This derivation can be written as follows. See, 3 round is to 1 wrinkled, and then 3 yellow is to 1 green. When these two combine together, we can get 9 round and yellow. 3 threes are 9. Round and yellow 9, and then wrinkled and yellow. 1 into 3, 3. So 3 wrinkled and yellow, and then 3 round and green. 3 multiplied by 1. 3 ones are 3, and then wrinkled green. So wrinkled green, 1 into 1, 1. So wrinkled green. So we got this ratio 9:3:3:1. Based on this dihybrid crosses. Now what is dihybrid crosses? Crosses between plants differing in 2 traits, 2 characters. He proposed this law of independent assortment. What is independent assortment? When 2 pairs of traits or characters are combined in a hybrid, the segregation of 1 pair of characters is independent of the other pair of characters. The 1 pair of characters, that is round and yellow, they separate independent of this wrinkled and round, and this round and wrinkled, they separate independent of this yellow and green. So the separation or the formation of gametes is independent of the other pair. So when 2 pairs of traits are combined in if 2 pairs are present in a hybrid, and the segregation of 1 pair of characters, they segregate independently, and the other pair they segregate independently. That is the law of independent assortment. This law of independent assortment can be easily studied by using this Punnett square. Already we studied about this Punnett square. The Punnett square can be used to understand the independent segregation of the 2 pairs of genes during meiosis and the production of egg and pollens in the F1 plant. F1 plants, they are capital R, small r, and capital Y, small y. First, consider the segregation of 1 pair of genes, capital R and R. Only these 2 pair of genes first we will see. 50% of the gametes have R. The 50, it will separate this 1 pair. We will take also these 2 pairs separately we will study. So 1 pair, if we take capital R and small r, then 50% of the gametes they have R gene and 50% they have small r. Now, besides each gamete having either R and capital R and small r, they should have another character because they differ in 2 characters. So another characters, genes also, they should have capital Y or small y. The important thing to remember here, is that segregation of 50% of R and 50% of small r is independent from the segregation of 50% of y and 50% of small y. So the separation, 50% of gametes will receive capital R and 50% of gametes will receive small r. And here in this separate pair, 50% of the gametes will receive this capital Y and 50% of the gametes will receive this small y factor, that is gene. Therefore, 50% of the R bearing gametes, they has Y and the 50%. So, 50% of the R bearing, therefore 50% R bearing gametes, they has capital Y and the 50% of the small y they will have. Similarly, 50% of the capital R bearing gametes, they has capital Y and the other 50% they have small y. Thus, there are 4 different genotypes of gametes we get. In this way, we get 4 different, all in the frequency of 1/4 of the total gametes produced. That is 25% of the total gametes produced. So, in this way, we got 4 types of gametes, capital R, capital Y and capital R, small r, capital Y, small r, small y. So, each in the frequency of 25% of the total gametes formed. So, in this way, by using this Punnett square, we can easily understand the formation of gametes and also the combination of gametes to form zygotes and also the F2 plants. So, in dihybrid cross, we got 2 different types of phenotypes and 9 different types of genotype. So, that's all for today, children. Tomorrow, we'll study about chromosomal theory of inheritance. Thank you, students.